Assalamu alaikum and welcome again to the Medical Concept with Dr. Falak. And today we are going to discuss about the white blood cells, their classification, function, and their genesis. By the end of this session, you all should be able to classify the different type of white blood cells. You should be able to describe the various function and properties of the white blood cells and explain the genesis of white blood cells. Now this slide shows the classification of the white blood cells based on cytoplasmic granules. Importantly, it is to note that all WBCs, they contain primary or azurophilic granules, which are actually lysosomes rich in enzymes like myeloperoxidase, elastase, and defensin. However, the classification into granulocyte and agranulocyte is actually based on the presence or absence of secondary or specific granules. So WBCs are actually divided into two main groups, that is the granulocyte and agranulocytes. Granulocytes have secondary specific granules. They are typically multi-lobe nuclei. They include neutrophil. Neutrophil are the first responder in the acute inflammation. Eosinophil, they are important in the parasitic infection and allergies. Then comes the basophil, that is involved in hypersensitivity reaction and histamine release. Then comes the agranulocytes. These lack secondary granules, though primary granules are still present and have simpler nuclei. They include monocyte, that are differentiated into macrophages and perform phagocytosis. Then comes the lymphocyte that are central to the adaptive immunity, including B and T cells. So the key point is that all WBCs have primary granules, but only granulocytes have secondary granules, which form the basis of this classification. Now this slide focuses on the neutrophils, the most abundant white blood cells that comprising of about 50 to 70% of the circulating white blood cells. They are called polymorphonuclear cells because of their multi-lobe nuclei, typically with 3 to 5 lobes with help in migration through the tissues. In terms of staining, you can see here that neutrophils are described as neutral staining cell. Their granules take up both acidic as well as basic dye, giving a pale lilac appearance on routine staining. Their cytoplasm contains faint secondary specific granules with antimicrobial substances like lactoferrin and lysozymes. Functionally, neutrophils act in the process of phagocytosis, and they are responsible for killing bacteria and the fungi. They release enzymes and antimicrobial substances, and they contribute to the pus formation at the site of infection. They play a major role in innate immunity, especially against bacterial infection, and they act as a first line of cellular defense. Now this slide highlights the eosinophils. These are actually the type of granulocyte comprising of about 1 to 4% of the total white blood cells. Morphologically, they are characterized by the bilobe nuclei and a large, coarse, uniform cytoplasmic granules. In terms of staining, eosinophils are described as acidophilic cells. Their granules stain intensely with acidic dyes like eosine, giving them a bright red or orange appearance. These specific granules, they contain potent substances such as major basic proteins and peroxidases, which are particularly toxic to the parasite, especially helminths. Functionally, eosinophils play a key role in the defense against the parasitic infection. They modulate allergic and inflammatory responses, including asthma. They phagocytose antigen-antibody complexes, and they release enzymes that contribute to the inflammation and allergy regulation. In immunity, their major role is in parasitic destruction, where they release toxic granules protein into their parasitic surface. Now this slide focuses on the basophils, the least abundant granulocytes, making of about 0.5 to 1% of the total white blood cells. Morphologically, basophils have a bilobe nuclei, or simply we can say S-shaped nuclei, which is often obscured by the large and coarse granules. In terms of staining, basophils are basophilic, meaning their granules stain intensely with the basic dyes, producing a deep blue or purple appearance. These granules contain important mediators such as histamine, heparin, and other inflammatory substances. Functionally, basophils, they release histamine and heparin. They play a role in allergic and inflammatory reactions, and they promote a vasodilatation and increase vascular permeability. In immunity, their role is mainly related to hypersensitivity reaction, where the release of histamine leads to vasodilatation and leakage of fluid from the blood vessels. This slide focuses on the lymphocytes, which are agranulocytes and the key cells of the immune system. Morphologically, lymphocytes are characterized by a large round nucleus that occupy most of the cell and only a thin rim of scanty cytoplasm. They lack prominent secondary granules, which is why they are also classified as agranulocytes. Functionally, lymphocytes are central to both innate and adaptive immunity. There are three major subdivisions. B lymphocytes or B cells, they're responsible for humoral immunity. They differentiate into the plasma cells that produce antibodies and memory B cells. Then comes the T lymphocyte. T lymphocyte, they are actually the mediator of the cellular immunity and they include further types, that is helper T cell, that is having a specific protein, that is CD4 on its surface. That's why they're also known as CD4 cells. Cytotoxic cells that are having CD8 protein on their surface. So that's why also known as CD8 cells, as well as regulatory T cells. Another type is the natural killer cells. They are forms the part of the innate immunity. They provide early defense by killing virus infected cells and the tumor cells without prior sensitization. The key takeaway is that lymphocytes are highly specialized agranulocytes with a dominant nucleus and they play a central role in a specific immune response and immunological memory. Now let's talk about the monocyte, the largest circulating white blood cells and an important agranulocyte. It has a characteristic kidney-shaped nucleus and abundant pale blue-gray cytoplasm that contain only primary or eosinophilic granules. They circulate in the blood for just 1 to 3 days before migrating into the tissue. Once they enter to the tissue, they differentiate into specialized cells such as macrophage, osteoclasts, and dendritic cells. Macrophages are powerful phagocytes that remove pathogens and dead cells. Osteoclasts are responsible for bone resorption and remodeling, while dendritic cells, they capture antigens and present them to T cells, thus link innate and adaptive immunity. In addition to the phagocytosis, monocytes secrete cytokines and functions as professional antigen-presenting cells, making them essential regulators of the body immune response. Now let's look at the normal distribution in the blood. The normal total white blood cell count is approximately 7,000 cells per microliter of the blood with a normal range of 4,000 to 11,000 cells per microliter. Neutrophils are the most abundant leukocytes making up about 62% of all white blood cells. Lymphocyte accounts for about 30%. Monocyte make up around 5.3%. Eosinophils account for 2.3%. Finally, basophils are the least common and only 0.4%. Remember these percentages as these are frequently tested in physiology and form the basis for interpreting the differential leukocyte count in the clinical practice. All white blood cells originate from hematopoietic stem cells in the bone marrow, but their maturation pathways differ. Neutrophils, eosinophils, basophils, and monocytes are produced and mature in the bone marrow before being released into the bloodstream. Interestingly, the bone marrow stores about 3 times more of these cells that are present in the blood, providing an approximately 6 days reserve. Lymphocytes also originate in the bone marrow. B cells and natural killer cells, they mature in the bone marrow, whereas the T cell migrate to the thymus and complete their maturation there. Afterward, all lymphocytes populate secondary lymphoid organs such as lymph node, spleen, and mucosa-associated lymphoid tissue, where they participate in immune responses. This diagram illustrates the process of hematopoiesis or the formation of blood cells in the bone marrow. It all begins with a multipotent hematopoietic stem cell which give rise to two major progenitor cells, the common myeloid progenitor and the common lymphoid progenitor. The common myeloid progenitor produce red blood cells, platelet, neutrophils, eosinophils, basophils, and the monocyte. During granulocyte development, the cell passes through several stages. Myeloblast, then we have promyelocyte, myelocyte, band cell, metamyelocyte, band cell, and finally become a mature granulocyte. Monocyte, after entering tissue, further differentiate into macrophages and the dendritic cells. The common lymphoid progenitor cells give rise to B cells, T cells, and natural killer cells. The B cell further differentiate into plasma cells, while T cell mature in the thymus before participating in adaptive immunity. As these cells mature, their nuclei become more condensed, specific granules appear, and they gradually lose their ability to divide, resulting in a fully functional blood cells ready to defend the body. Leukopoiesis is regulated by means of various factors, for example, hematopoietic growth factors, colony stimulating factors, various hormones, interleukins, cytokines, and also overall bone marrow microenvironment. The lifespan of white blood cells varies depending on the cell type. Neutrophils are the shortest lived, circulate in the blood for about 6 to 8 hours before entering the tissues, where they survive for 1 to 3 days. Eosinophils and basophils, they also have relatively shorter lifespan. Monocytes circulate for 1 to 3 days before migrating into the tissue, where they differentiate into macrophage or dendritic cell. In contrast, macrophages are long-lived tissue cells that can survive for months to years. Unlike innate immune cells, adaptive immune cells are generally long-lived and can develop immunological memory. Naive B cells and T cells can survive for weeks to months while continuously circulate through the blood, lymph, and lymphoid organs. After encountering an antigen, they become activate and differentiate into effector cells, which are short-lived and survive for only 1 day to weeks. A small proportion then become memory lymphocyte, which can persist for years or even a lifetime, providing a long-term immunity and a faster response upon re-exposure to the same antigen. Now coming to the various characteristics of white blood cells. One and the most important is the maturity. This is especially true for monocyte or the macrophages. Monocyte in the blood streams are immature. They migrate into the tissue, undergo a five-fold increase in the size. They become powerful macrophages and develop enhanced phagocytic capabilities that enable them to effectively combat infections and to clear pathogens. Second most important property of the white blood cells is the diapedesis. And what is diapedesis? Diapedesis is the passage of the white blood cells by squeezing through the gap between endothelial cells of the blood capillaries and the post-capillary venules to enter the tissue. Sometimes this migration is transcellular also. The characteristic of white blood cell is their amoeboid motion. Chemotaxis is the directed movement of the white blood cells toward a chemical signal or site of infection. It depends on the concentration gradient of the chemotactic substance that is greatest near source, and most often it is unidirectional. The chemical substance can include bacterial or viral toxins, degenerative product of inflamed tissues, reaction product of complement complexes, or reaction product caused by plasma clotting in the inflamed areas. After a pathogen is recognized, the phagocyte extends pseudopores around it and engulfs it, forming a membrane-bound vesicle that is known as phagosome. If the microbe is coated with antibodies or the complement protein, the process becomes even more efficient through receptor-mediated recognition, a phenomenon that is known as opsonization. The phagosomes then fuse with a lysosome to form a phagolysosome. Inside this compartment, lysosomal enzymes, reactive oxygen species, and other antimicrobial substances they digest and destroy the microorganisms. Finally, any undigestible debris is expelled from the cell by the process of exocytosis, completing the process of phagocytosis. Both neutrophil and macrophages they possess powerful mechanism to destroy invading microorganisms after phagocytosis. First, lysosome fuses with the phagosome and release proteolytic enzyme that digest pathogens. They also contain potential bactericidal substances that kill microbes even before enzymatic digestion is complete. During the respiratory burst, these cells generate highly reactive oxygen species, including superoxide, hydrogen peroxide, and hydroxyl radicals, which rapidly destroy bacteria. In neutrophils, the enzyme myeloperoxidase convert hydrogen peroxidase into hypochlorite or bleach, one of the most effective antimicrobial agents produced by the body. Macrophages have additional advantage. Their lysosome contain lipases that help digest lipid-rich bacterial cell wall, such as those of Mycobacterial tuberculosis. Together, these mechanisms make neutrophil and macrophages highly effective defender against the infection. Let's talk about the reticuloendothelial system, or RES, also known today as the mononuclear phagocyte system. This system is made up of circulating monocyte, tissue macrophages, and specialized phagocytic cells that protect our body by removing harmful substances. The process begins with monocytes circulating in the bloodstream. Whenever there is tissue injury or infection, these monocytes leave the blood vessel, enter the tissue, and differentiate into macrophages. These macrophages can either remain mobile or become fixed tissue macrophages. The primary function is phagocytosis. They engulf and destroy microorganisms, toxins, dead cells, cellular debris, and other unwanted particles that helps maintain tissue health and support immune defense. Different tissue contain specialized macrophages with unique names. For example, in the liver, they are called Kupffer cells. In the lung, they are known as alveolar macrophages. In the brain, microglia. In the skin, Langerhans cells, while the spleen and the lymph node also contain specialized macrophages responsible for filtering blood and the lymph. Overall, the reticuloendothelial system serves as the body professional clean-up and defense network, playing a vital role in both innate and tissue homeostasis. So this was the end of today's video, and I hope you found this video helpful. If you enjoyed this session, don't forget to like, share and subscribe to Medical Concept with Dr. Falak for more easy to understand medical lectures. See you soon in the next video. Allah Hafiz.